Well, hello students. I hope you all are good and you are doing well. In this particular video lecture, we are going to talk about the bacterial appendages. Okay. Now, what are appendages? First question. Your hand and your feet or legs are appendages, right? The tentacles of Hydra, they are appendages. Okay. Or wings of bird, they are appendages. Okay. So what appendages are? Appendages are the structures of an organism which help them in locomotion, help them in movement, help them in food capture, help them in offense defense. Okay. So all these structures are called appendages. Okay. Now in case of bacteria, which is an unicellular organism, what could be the appendages? Well, now in case of bacteria or the prokaryotic cells, the appendages are flagellum and pili. There are only two things, flagella and pili. Now, flagella is plural, flagellum is singular. Okay, so any uh, eukaryotic organism has these two appendages, or I should say they may or may not have these two appendages. If they have appendages, they at max can be these two, flagella and pili. Okay, great. Now, in our previous lecture, we have already seen the bifurcation or different types of bacteria on the basis of flagella or flagellum, right? We have seen atrichus, we have seen the monotrichus, we have seen the amphitrichus, we have seen lophotrichus, cephalotrichus, peritrichus, right? We saw all different types of bacteria on the basis of distribution of flagella on their body or on their cell, correct? Now, what we are going to see in this lecture is the structure of flagella, okay? The function of flagella and pili. So let's start. Now, flagella, okay? When we are talking about flagella, flagella can be found in both eukaryotic cells as well as in prokaryotic cells. That is, they can be found in both eukaryotic and prokaryotic organisms. So flagella is found in both. Now, what is the function of flagella? Again, it is a whip-like structure which helps in locomotion, food capture, and offense and defense. Okay, so whether it is in eukaryotic organism or in prokaryotic organism, the flagella is always like a whip. Okay, and its role is that it is going to help in movement and locomotion, food capture, okay, or in offense defense. Correct. Now let's see that, or note that this flagella has same function and almost same appearance. I'm not saying structure, I'm saying appearance, right? Same appearance and same function in both eukaryotes and prokaryotes. Does that mean they are same? No, they are not. Okay. See, the bacterial flagella, structurally, the bacterial flagella and the eukaryotic flagella are not, okay. So structurally as well as chemically, the eukaryotic flagella and prokaryotic flagella are different. They are not same. Why so? See, structure we will see here of bacterial flagella. Eukaryotic flagella we will talk in some other chapter. However, if we talk chemically, the prokaryotic or bacterial flagella is made up of flagellin protein, whereas the eukaryotic flagella is made up of tubulin protein, or you sometimes say microtubule. Okay, actually it is made up of microtubules, but microtubules in turn are made up of tubulin protein. Correct. So chemically, the bacterial flagella is made up of flagellin, and eukaryotic flagella is made up of tubulin. Remember it. It is important. Okay, great. So flagellin and tubulin. Prokaryotic or bacterial flagellum made up of flagellin. Now, let's look at the structure of a bacterial flagella or prokaryotic flagella. How they appear, what is the structure, right? So let me draw it for you. So the bacterial or prokaryotic flagella or flagellum. Let's see. Now, to draw it, let me first make the cell membrane and the cell wall, right? So this is cell membrane and the other one is cell wall, correct? And the space between the two, is called periplasmic space. Okay, great. Now, there is a shaft like structure, okay, running into the cell wall and cell membrane through the periplasmic space, correct? Now, when you see this red structure, the shaft has two rings in the cell membrane, okay, and similarly there are other two rings in the cell wall. Okay, great. Now, apart from this, can you see there is a curved perpendicular structure and then this. Now, this blue part, this elongated whip-like part is called filament. This is the filament of the flagella, okay, or flagellum. And this filament is made up of flagellin protein, okay. It is made up of flagellin protein and it is made up of 4+4 arrangement of flagellin protein, okay. Now, what is this 4+4 is not important here. But this filament is in turn made up of 8 thread-like structures which are made up of flagellin protein. So these flagellin proteins are like your hairs, okay? These are filamentous structures and 4+4 yani ki 8 flagellin filaments are found or they combine to form this filament. Okay, great. Now, this curved structure is called hook. The curved structure is called hook and it is partially embedded into the cell wall. So hook is partially embedded into the cell wall and it is a curved structure. On the other end, what you find is filament. Okay, great. Now, when we see the portion of flagellum which is inside the cell wall and cell membrane, what we find is it has 4 rings, okay. The upper ring in the cell wall region is called L ring, the lower one in the cell wall is called T ring, okay. The upper ring in the cell membrane is called S ring and the lower ring in the cell membrane area is called M ring. Remember this. So now this entire structure, all these 4 rings and the red structure which is called shaft are combinedly called basal body. These are combinedly called basal body. Okay. So see, the bacterial or prokaryotic flagellum has 3 distinct parts. First one is basal body that remains embedded into the cell wall and the cell membrane. Then there is a curved hook. And finally, on the distal end, there is filament. Now, the filament is the longest part. Okay. So, if we are talking about bacterial flagellum, it has 3 parts: basal body, hook, and filament. Filament, you have seen, hook, you have seen. The basal body is composed of a shaft and 4 rings, and these 4 rings are L, P, S and M, out of which L and P are found in the cell wall and S and M are found in the cell membrane. Okay. Now what happens? This hook rotates either in the clockwise or anti-clockwise. Okay. So this hook rotates. As a result, the entire flagellum rotates. And when this flagellum or the filament of the flagellum rotates, it gives a undulating movement, okay, like a spiral movement. As a result, this produces a force, and using this force the bacteria swims in a medium, or we can say it shows locomotion or movement. Okay, So this is how flagellum works. Great. So this is about the bacterial or prokaryotic flagellum, the structure of prokaryotic flagellum. Now let's see the second bacterial appendage, that is pili. Okay. Now what is pili? These are small hair-like structures, okay, which are found all over the bacteria, that is on the outer surface of the bacteria, as you can see here. So on a bacterium, I'm drawing these small hair-like structures. These are pili. Okay. So these are pili. Great. Now just remember that pili is plural form, whereas pilus is the singular. Okay. Now, so what we have seen here is the pili are small hair-like structures. They are found all over the bacterial surface or outer surface, and pilus is singular, pili is plural. Now, apart from that, let me tell you that this pili are hollow tube-like structures, right? They are hollow tube-like structures. So let's see the structure of it. Okay, so see, it is a hollow tube-like structure. They are thin and short tube-like structure. Okay, now what they are made up of? Again, they are made up of protein. Which protein? They are made up of pyline protein. Okay, they are made up of pyline protein, and the thing to remember here again is pyline protein is non-contractile protein. What do we mean by non-contractile protein? See, your muscles, right? The muscles are made up of proteins, but these proteins are contractile, as in they can contract, that is, they can change their length, correct? Whereas pyline is non-contractile, so they cannot change their length. Now, most of the proteins are non-contractile. Okay, great. So, pili is a short, thin, tube-like structure present all over the body of bacteria, and it is made up of a non-contractile pyline protein. One more thing, let me tell you that it is not necessary that each and every bacteria should have pili. They may not. Okay. Another important thing, when you will study cell, you will find cilia. Okay. Now cilia is similar to pili, but cilia is only found in eukaryotic cells. They are never found in prokaryotic cells. Okay. Do remember this. So bacteria can never have cilia. They can have pili or flagella. They cannot have cilia. Okay, cilia is a eukaryotic structure. Great. Now, if we take pili, a pili can be divided into two, that is shorter pili and longer pili. What are these shorter pili? Shorter pili are also known as fimbriae or infective pili, right? So we are calling this shorter pili either fimbriae or infective pili. Why are we saying it infective pili? Because it is found in pathogenic bacteria, or the bacteria which causes disease, which infect us, the bacteria which are infective to us, and that's why this pili is called infective pili. Okay, in most of the pathogenic bacterial strain which are infective to us and known to have pili, they uh, have this pili, shorter pili. Okay, and that's why we call them as infective pili. What they do here is they help in attachment. Okay, attachment to the host or substrate. So these small pili, shorter pili, will help this bacteria to adhere to a surface or to the host cell. Okay, so do remember this, fimbriae or infective pili. Apart from this, there is another type of pili, that is longer pili, correct? And what is longer pili? If you see longer pili, it is called F pili or fertility pili. Okay, now why we call it? Because it helps in sexual reproduction. Okay, and hence this pili is also known as sex pili. how it helps in sexual reproduction, or sexual reproduction does even takes place in bacteria? Yes, but it is quite different from what you know about sexual reproduction. Okay. Now, in bacteria, there is a phenomena called conjugation. And now, during conjugation, the genetic material is exchanged between the two partners. Now, wherever there is involvement of different genetic material, as a result, genetic variation comes in, we call such reproduction as sexual. Okay, and in conjugation, this exchange of genetic material takes place, and hence it is considered as sexual reproduction, one of the mode of sexual reproduction. Okay, and sex pili helps in conjugation. That's why it is helping in sexual reproduction. Sex pili or alternative pili or F factor. Okay, so it helps in conjugation, a type of sexual reproduction. Okay, great. Now see, the bacteria with F pili, okay, is considered as male or F positive or donor bacteria. Okay, now how does it look? Suppose the white one that I'm drawing is a bacteria, and this bacteria, you can see here, has a small round DNA that is plasmid, and this plasmid contains the gene for making this longer pili. Now, using this longer pili, this bacteria attaches with another bacteria. Okay, this with this longer pili or F pili, it attaches to another bacteria. Now, this bacteria does not have this extra plasmid. As a result, it cannot make longer pili or sex pili, and hence this bacteria is considered as female bacteria or recipient bacteria or F negative bacteria. Now, when these two bacteria gets connected with this sex pili, what happens is this extra DNA or extra plasmid, which contains the information for making F pili or sex pili, is transferred from this male bacterium to the F negative or female bacteria. So again, here the genetic transfer or the mixing of genetic material is taking place, and that's why it is a form of sexual reproduction. Okay, now see. So later what is happening? Later, the F negative or female bacterium is getting converted into male or F positive or donor bacteria. Now, this whole process is called conjugation. We will see this in detail. Okay, so as of now, just understand the role of longer pili or sex pili or F pili. What are they? Okay, great. Well, so these are the two bacterial appendages, their structure, okay, and their function and their type. I hope you got it. So do remember that bacteria cannot have cilia. Any prokaryotic organism do not have cilia. As appendage, they have this flagellum, which is prokaryotic type or bacterial flagellum, and pili. Is it okay? Great. So this is it for this lecture. In the coming lectures, we will see the detailed structure of bacteria or prokaryotic organism, and in detail we will start talking about individual components. That is, we will talk about the cell membrane, the cell wall, different types of bacteria on the basis of cell wall. We will talk about the glycocalyx, and in that we will talk about slime layer and the capsule. We will talk about the genetic material, about the plasmid. We will talk about ribosomes. We will talk about granules and things like that. So in detail, we will see the structure of bacteria. We will also see the gram staining. Okay? So on how we classify bacteria as gram positive and gram negative on the basis of gram staining. So these all things we are going to cover in the next video lecture. So keep watching these videos. Okay, stay tuned and stay safe. Thank you.